Welcome to the final session on human reproduction. In this class, we are going to discuss topics like fertilization, pregnancy, embryonic development, and also parturition. First of all, what is fertilization? You know, fertilization is the process of fusion of a sperm with an ovum, and it is happening within the fallopian tube. You know that during coitus or sexual intercourse, the seminal fluid will be released by the penis and it will be reaching the vaginal portion. This is called insemination. And after insemination, you can see the sperm cells are moving with the help of their tail. They can reach the junction of isthmus and ampulla. We can call it as the ambulary isthmic junction. At the same time, ovum that is released from the ovary will also reach there. You know, it is actually the secondary oocyte reaching here. It will be a premature ovum. So, both the sperm cells as well as the ova, they have to reach here in this junction called ambulary isthmic junction. Then only there will be process called fertilization. You can see that ovum is having different membranes. The different membranes covering the ovum are, first of all, there is a non-cellular layer called zona pellucida. You can see here, this is zona pellucida. And outer to zona pellucida, you can see a layer of follicular cells, and this layer is called corona radiata. In this diagram, you can see the sperm cells are reaching on the surface of the ovum in order to penetrate into the ovum. After penetration, there will be activation of the ovum. What is activation? Activation means all the events that will happen within ovum after the entry of the sperm cell. You know that only one sperm cell will be able to enter into the ovum, or there will be only monospermy. Polyspermy is prevented in the case of human beings. How it is prevented? The polyspermy will be prevented due to some changes that are happening within the ovum after the sperm entry. We will now go through the different steps or events happening after sperm entry. After sperm entry, first of all, there will be depolarization of the oolemma. This is fast block to polyspermy. And soon after this, there will be a cortical reaction, and this cortical reaction will result in formation of a membrane called fertilization membrane. So once fertilization membrane is formed, no other sperm cells can enter into the ovum. So only one sperm cell will be able to enter into the ooplasm. So thereby polyspermy can be prevented. So cortical reaction is considered to be a sure block to polyspermy. Now, soon after the entry of the sperm cell, what happens? You know that the cell is actually in a secondary oocyte stage and the cell cycle is being arrested in metaphase 2. Now after the sperm entry, there will be completion of the second meiotic division, and there will be release of a polar body. The second polar body will be released, and now the cell is getting transformed into ovum. Now we are going to discuss about the sex determination in humans. In human beings, male individuals are having chromosome pattern shown here, 44 autosomes plus XY condition. The sex chromosomes are X chromosome and one Y chromosome. Then in the case of female, the chromosome pattern will be 44 autosomes and 2 X chromosomes. So this is the chromosome pattern. In this diagram, you can see how sex determination occurs in human beings. You can see the parents here. Parents are having the chromosome pattern that I mentioned just now. And what happens here? You can see from the male individual, which is marked here, there will be formation of two types of sperm cells. One type of sperm cell will have X chromosome in it, and the other type will have Y chromosome. So we can say that 50% of the gametes that are produced from the male will carry X chromosome, and the remaining 50% of the sperm cells will carry Y chromosome as shown here. And what about the female? The female will be able to produce only one type of gamete or only one type of ovum, and that ovum will carry an X chromosome. So you can see it here. Okay, now you can see what happens. Suppose the sperm cell with X chromosome is binding with or entering into the ovum containing X chromosome. There will be formation of a female individual like this. You know, the chromosome pattern of females will be 2X chromosomes. What happens when a sperm cell with Y chromosome enters into an ovum? You can see it over here. The result will be a male individual, because male individuals, they have the chromosome pattern of XY. You can see the formation of the gametes. We can clearly say that the sex determination in humans, it is mainly controlled by the male individual. The male individual is responsible for sex determination in human beings. Now we are moving to our next topic, that is cleavage. What is cleavage? Cleavage is a process of mitotic divisions that are happening within zygote. So after the formation of a zygote, there will be repeated mitotic divisions happening within the zygote. You can see that zygote is a single cell. It is getting transformed into a 2-cell stage, then a 4-cell stage here, and further it will convert into 8-cell, then 16-cell, so it goes on. And cells that are formed as a result of cleavage, they are called blastomeres. So blastomeres, their number will increase as the cleavage proceeds. The embryo having 8 or 16 blastomeres in it is known as the morula. You may notice in this diagram that there is no considerable increase in the size of the embryo from the zygote or from the zygote to the morula stage. Why? Why there is no increase in size? In the 2-cell stage as well as in the 16-cell stage, the size of the embryo remains the same. Why? It is because the zona pellucida is remaining intact up to the morula stage. You know, zona pellucida is formed before ovulation, right when the secondary oocyte was in the follicle, Graafian follicle. There is formation of the zona pellucida. So this zona pellucida is remaining intact even up to the morula stage. That is why there is no considerable increase in the size of the embryo. You can see it here. Now, what happens to morula? Morula further develops into a blastula. Blastula is an embryonic stage with a structure like this. Here we can see a sectional view of blastula, and in this sectional view you can see the outer layer of cells are called trophoblasts, and the inner cell mass is also there. There are some cells attached on to the trophoblast, that is called inner cell mass. You know, the trophoblast is the layer which is involved in formation of fetal membranes. And what about the inner cell mass? Actually, from this inner cell mass, the actual embryo is formed. So here, blastula is having two regions, the outer layer of trophoblast and a group of cells called inner cell mass. The mammalian blastula is known as blastocyst, and blastocyst is the embryonic structure which is undergoing implantation. You know, after fertilization, it will take around 7 days for implantation or for attachment of the embryo to the uterine wall. So this is a diagram of blastula given in detail. You can see the endometrium over there. So this is endometrium. You can see the inner cell mass, trophoblast, and also there is a cavity labeled here. This cavity over here, that is called the blastocyst cavity. Now what is implantation? Implantation is not merely an attachment of the blastocyst on uterine wall. After attachment, the uterine cells divide rapidly and covers the blastocyst, and as a result, the blastocyst becomes embedded in the endometrium of the uterus, and this is known as implantation. So remember, only when the blastocyst is getting embedded within the endometrial wall, we can call it as implantation. So in this diagram, you can see what happens after fertilization. After fertilization, day 1, day 2, day 3, all these days are passing, and finally the embryo, which is moving through the fallopian tube towards the uterus, it is moving towards the uterus or reaching uterine cavity, and you can see the blastocyst on day 7, it undergoes implantation. The trophoblast layer of the blastocyst is getting attached with the uterine wall, and that is called implantation. You can see it here, and further this embryo will develop within the wall of uterus. And you know, the development of the embryo within the wall of uterus is called pregnancy. Pregnancy, in the case of human beings, it will last for around 9 months. So you can see in this diagram an embryo. An embryo is connected with the uterine wall by way of an umbilical cord. You can see it over here. This is umbilical cord, and you can see a yolk sac, and here this portion is being labeled as the uterine cavity or cavity of uterus. So the cavity is getting reduced as the embryo is undergoing development. Okay, so this portion, you can see the plug of mucus in the cervix. This is cervical plug here. Now, after implantation, what is the next step? After implantation, you can see some finger-like structures will arise from the portion of the embryo, and these finger-like structures are named as chorionic villi. You know, chorionic villi, further, it will be surrounded by the uterine tissue as well as the maternal blood. And now, the chorionic villi and uterine tissue, they become interdigitated with each other. I repeat, they become interdigitated with each other, and it will result in a structural and functional unit of the development, and we call it as placenta. So here you can see the placenta is formed here in this portion. You can see it here. Okay, the chorionic villi first formed, and further it is resulting in the formation of a placenta. Now, what are the functions of placenta? Placenta is performing different functions in the case of human beings. The first function will be facilitating the supply of oxygen and also the nutrients, because the placenta will act as a mediator now. So here it is also helping for the removal of carbon dioxide as well as waste materials. So all the waste materials that are produced within the embryo or in the developing fetus will be removed by the help of placenta. Now, placenta, you know how it happens? It happens because there is an umbilical cord connecting the embryo with placenta. Placenta can also act as an endocrine tissue because it is producing a set of hormones. What are the different hormones being produced by placenta? The hormones are human chorionic gonadotropin or HCG, human placental lactogen, we can call it as HPL, then estrogens, progesterone. So all these are the different hormones being produced by placenta. That is why we say that placenta is having an endocrine role. In the later phase of pregnancy, we can also see relaxin being produced from the ovary. Remember, there are three main hormones produced only during pregnancy, and they are HCG, HPL and relaxin. In the case of estrogen, progesterone, they can be produced even without a pregnancy. So at this point, the hormones, the three hormones produced only during pregnancy are HCG, HPL and relaxin. Immediately after implantation, you can see the inner cell mass is differentiating itself into the germ layers. There are three germ layers formed in the case of human beings, and they are ectoderm, endoderm, and in between the ectoderm and endoderm there is a mesoderm. These three germ layers are formed in the stage of embryo called gastrula. You can see in this diagram the cleavage. As the cleavage proceeds, the 8-cell stage getting converted into a blastula. Then blastula, a cross section is given there. The blastula is having blastocyst. Further, it undergoes gastrulation. Gastrulation involves a series of morphogenetic movements, and due to these morphogenetic movements, you can see there indicated in the form of arrows. So due to the morphogenetic movements, this structure, the blastula, is undergoing a differentiation to form a gastrula. And in gastrula you can see the ectoderm, endoderm. So all the structures are there developing within the gastrula. So the inner cell mass is getting differentiated into the three germ layers. Now we go to the next topic, that is embryonic development. Embryonic development in human beings will occur within the uterus, and it will happen for 9 months. And these 9 months can be divided into 3 trimesters. By trimester we mean 3 months. So the first trimester, by using that word, we mean the first, second and the third month of pregnancy. And what is the second trimester? Second trimester, by that we mean the 4th, 5th and 6th month of pregnancy. And the final trimester or the third trimester, that involves 7th, 8th and the 9th month. So the last 3 months will form the third trimester. So there are 3 trimesters in the case of a normal embryonic development. You can see in this diagram the embryo develops into a fetus, and fetus is undergoing lot of changes to form a final stage over here. You can see different structures are developing within the fetus. Here a table is given for you. From this table you can clearly understand the main features developed during different times. The embryonic heart will develop in the first month of pregnancy. The doctors, with the help of a stethoscope, they can hear the heartbeat in the first month itself. Okay. Is it interesting? Now what happens next? Limbs and digits. Limbs and digits, even though they are rudimentary, they will be developed in the second month. Then external genital organs. If it is a male, testis, and if it is a female, a pair of ovaries will be developed after the completion of the first trimester. Then the first fetal movements. The mother will be able to identify that her baby is moving within the womb or within the uterus. That can be sensed during the 5th month. And also in this 5th month, there will be development of hair on head. We cannot see that, but the mother is able to identify the fetal movements here in the 5th month itself. Now what happens? Fine hair developed around the body, eyelids will develop, eyelashes will also develop. So that will happen after the completion of the 2nd trimester. Now we come to the final part of human reproduction, that is parturition. What is parturition? Parturition is the expulsion of a fully developed fetus from the uterus. Parturition is actually induced by a complex neuroendocrine mechanism. By neuroendocrine we mean there is involvement of the nervous system as well as a set of hormones in it. The signal for parturition will originate from a fully developed fetus and also from a fully matured placenta. So a fully developed fetus and placenta will produce some factors, and that will act as a signal for parturition. And these factors will cause some mild uterine contractions. Remember, these contractions are very mild uterine contractions. And we can call these mild uterine contractions as fetal ejection reflex. So this is very important. In the beginning there will be fetal ejection reflex. Further, this fetal ejection reflex will trigger the release of oxytocin from maternal pituitary. What is maternal pituitary? The pituitary gland of the mother is known as maternal pituitary. So from maternal pituitary there will be release of oxytocin. And what happens next? This oxytocin will act on uterine muscles, and now the uterine muscles will undergo vigorous contractions. And when uterine muscles undergo vigorous contractions, it will again stimulate the pituitary, maternal pituitary, to release more and more oxytocin. So this will happen as a method of a positive feedback mechanism. You know what is positive feedback mechanism? Here, as and when the contractions are going stronger and stronger, there will be release of more and more oxytocin from maternal pituitary. So this will happen in a cyclic manner, contractions becoming stronger and stronger. At the same time, the production of oxytocin becomes high, and that will lead to the expulsion of the embryo from the uterus. So this will happen, as I told you in the last session, this will happen through a birth canal. So this event is called parturition. So remember, parturition is induced by a fetal ejection reflex, and also there is involvement of oxytocin in parturition. Now we go to the next topic, that is lactation. What is lactation? Lactation is done by the female. It is the nourishment of the young one by the milk produced from the mammary gland. You know, during the initial phase of lactation, the mother will produce a yellow colored milk, and we call it as colostrum. And this colostrum is rich in antibodies, and these antibodies will give a passive immunity to the newborn. What is passive immunity? Immunity attained by direct introduction of antibody is known as passive immunity. So here the newborn will have a weak immune system, so it is more prone to infections which will come via the respiratory tract or digestive tract. So here colostrum will provide all the necessary antibodies which will help them to prevent such infections from inhaled or ingested pathogens. That is why doctors will say that colostrum is very important. The feeding of the young one, especially during the initial days after parturition, is very important. It is because it will give a passive immunity to the newborn. Do you know which antibody is present in colostrum? Within colostrum, there is antibody A or immunoglobulin A. So, a young one should be nourished well during the period after parturition. So with that we conclude our session of human reproduction. Now we will answer a set of questions based on these topics. Here is your first question. Match column A and column B with respect to the development of embryo and appearance of features. Can you answer this question? In column A or in the first column, some features are given, or in column B or in the second column, some durations are given. I know you can answer this question easily. As I told you, the embryonic heart will develop in the first month. Limbs and digits will develop in the second month. External genital organs, you can match here with the first trimester. By the completion of the first trimester, there will be development of external genital organs. First fetal movements, you know the answer. First fetal movements will occur in the 5th month. Then finally, the development of hair, eyelids are separating and eyelashes will develop in the, yes, you know that it will happen after the 2nd trimester. So now you have the answer, that is option C. Option C will be our answer for this question. Second question, choose the option showing the correct labeling for the following diagram in the sequence A, B, C and D. So a diagram of embryo developing within the uterus is given here. Here is a set of options. Can you answer this question? Yes, it's very easy. Here the portion labeled as A is umbilical cord. The portion labeled as B, what is that? Yes, they are placental villi. Then the portion labeled as C, that is a yolk sac, and D, that is the cervical plug. So you know the answer, that is option A. Now we move on to the third question. The correct sequence in embryonic development is, which is the correct sequence? First of all, there will be fertilization, and after fertilization there is formation of a zygote, and after formation of a zygote it will undergo cleavage, repeated mitotic divisions to form a morula. Morula is the 16 cell stage, and further morula transforms into a blastula. Before developing into a gastrula, it will develop into a blastula, and blastula will have around 32 cells in it. So here our answer will be option A. Now coming to question number 4, it is about polyspermy. Polyspermy during fertilization is prevented by, options are release of acrosomal enzymes, option B, formation of fertilization membrane, C, enzymes produced by glands on oviductal wall, option D, removal of fertilization membrane, and E, capacitation. You know, polyspermy is entry of more than one sperms into an ovum, but it will be prevented by some reactions happening on the ulama, as well as some changes happening within the cytoplasm of the ovum after entry of the sperm. And altogether we can call it as activation of the ovum. I told you in the beginning there will be different reactions like depolarization of the ulama, and also there will be cortical reaction which results in fertilization membrane. So here the answer will be option B, formation of fertilization membrane. It is a sure block to polyspermy. Here by release of acrosomal enzymes, it will only help in penetration of the sperm cell. Now what about option C, enzymes produced by glands on oviductal wall? It is not our answer. Now coming to removal of fertilization, no, it is actually formation of fertilization membrane. So D is wrong. Capacitation, you know that capacitation is the activation of sperms. So you have the answer B. Now match the following. Can you match column A with column B? What is first menstruation? First menstruation will happen when the individual attains puberty, and this is called menarche. So 1 you can match with menarche. What about 2, preventing polyspermy? You can match with fertilization membrane. Fertilizing is a substance which will be covering unfertilized egg, and it is helping for the species specificity of fertilization. And here on the surface of a sperm cell, it will be covered with a corresponding anti-fertilizing. So here fertilizing you can match with unfertilized egg. Fusion of pronuclei. What is that? You know, the male pronucleus and the female pronucleus will undergo a fusion, and we call it as amphimixis. Amphimixis is actually the completion of fertilization. Now the fifth one, the first fecal matter. First fecal matter produced by a newborn is known as meconium. Meconium is black colored. So here our answer will be option B. Coming to question number 6, identify the timing of release of second polar body. Here is a set of options. You know, second polar body will be released only after the sperm entry. Sperm is actually entering into a secondary oocyte, and after the sperm entry there will be completion of second meiosis. Then there will be release of the second polar body. So here you can pick the answer. The answer will be option A, because it is happening after the sperm entry, okay, and it is just before amphimixis. You know what is amphimixis? Yes, it is a fusion of the two pronuclei, the male pronucleus and the female pronucleus. So here our answer will be option A. Now coming to question number 7. The hormones that are produced during pregnancy are only during pregnancy. Yeah. The options are estrogen, HPL, HCG. Option B, HCG, HPL, progesterone. C, progesterone, HCG, HPL. D, progesterone, relaxin, HCG, HPL. Then comes option E, HCG, HPL, relaxin. Is it a difficult one? No, it's very simple. Hormones that are produced during pregnancy, they are HCG, HPL and relaxin. Because HCG and HPL, they are being produced from the placenta. And relaxin is produced from ovary, and it will happen only during pregnancy. Here, the other hormones like estrogen, progesterone, they are being produced even before the pregnancy or even without a pregnant situation. So here, answer will be option E. Now coming to question number 8, you have a diagram. Observe this diagram and try to identify the correct labeling. Identify X and Y in this diagram. Here is a set of options. You know, this is the diagram of blastocyst. The outer layer is being labeled as Y and a group of cells being labeled as X here. So what is X here? The X here, that is the inner cell mass, and Y here, that is outer layer. What is the Y? It is the trophoblasts. So the correct answer will be option A. Now coming to question number 9, which of the following is wrong regarding parturition? Some statements are given. Here is a set of statements. Option A, signals for parturition originate from a fully developed fetus and placenta. That is a correct statement. Oxytocin is released from fetal pituitary as a result of fetal ejection reflex. You know, this is a wrong statement. Why? You know why? It is because actually from maternal pituitary, oxytocin will be released due to fetal ejection reflex. So this is a wrong statement. Now coming to other options, parturition is induced by a complex neuroendocrine mechanism. That is a correct statement. Oxytocin acts on uterine muscle and causes stronger uterine contractions. That is also true. Mild uterine contractions are associated with the fetal ejection reflex. So that is also true. So here we have only one wrong statement. That is option B. So the correct answer is option B. Now coming to the last question of this session, in cleavage up to morula stage, some statements are given here. You have to identify the correct statements. Remember, here in this question they are asking about embryonic development from zygote up to this morula stage. We can go through the options now. Size of blastomeres will decrease as their number increases. This is true because the number of blastomeres will go on increasing. At the same time, the embryonic size will remain the same. So the size of blastomeres will be decreasing. The size of the blastomere in the 2-cell stage will be larger than that is present in an 8-cell stage or 16-cell stage. So this is a correct statement. DNA synthesis does not occur. This is wrong. DNA synthesis will occur during cleavage because it is mitotic division. Size of embryo remains the same as cleavage progresses. This is a correct statement. As I told you, here the zona pellucida will remain intact up to morula stage. So there will be no increase in the size of the embryo up to morula. Now coming to statement number 4, the size of embryo increases as the cleavage proceeds from zygote to morula. This is a wrong statement. Here the correct statements are statement number 1 and statement number 3. So the answer will be option A. So with that, we conclude our session on human reproduction. In this last session, we discussed in detail the different events like fertilization, pregnancy, embryonic development, parturition and lactation. Thank you.